Thanks so much. And thanks so much to Marisa and Michelle. This is a fantastic symposium, and I'm delighted to be part of it. So today I'll be telling you a little bit about biobanking. I'll try and get us a little bit back on track. So I'll talk quickly, but perhaps in the panel discussion, additional questions can be asked. So I have no disclosures. So the Brain Tumor Center Biorepository and Biomarker Core, or Tissue Core is commonly referred to, has a goal to maximize the translational impact of research on both brain and spinal cord tumors. And that certainly includes metastases as well. And so we really divide our activities up into two main groups, and I'll sort of walk you through some of the activities that we do within that. The first is what you might think of, and that's what both Harish and Hugo and others have talked about, and that's the acquisition, optimal storage, processing, and distribution of biospecimens from the operating room or from the clinic. And so I'll go through some of the activities associated with that and sort of explain to people what we do associated with that. A second important part, I think, of maximizing our translational ability is the analysis of some of these samples. Obviously, you've heard just fantastic talks on some of the really cutting edge technologies that can be applied to analysis of these tissues. And hopefully I'll convince you that some of the other maybe old school or older techniques may also be helpful in terms of confirming some of the findings that you find with these new techniques. And also additional advanced techniques that we have as well. And I should mention that we are a CLIA certified laboratory, so we do both clinical testing for the pathology department as well as work with clinical trials for neuro-oncology and neurosurgery, as well as our translational and basic science research. So sort of thinking about how we divide our activities, I'll talk first about biospecimen acquisition. So many of you are familiar with this process. So a patient is consented preoperatively, and in the operating room, we have just really talented and fantastic staff who are present there, who can answer questions from the surgeon, who can sort of be the in-between between the researchers and the investigators and the surgeon in terms of what is needed, and making sure, obviously, the number one priority is always tissue for clinical diagnosis and molecular stratification. And then any excess tissue can be acquired by the biorepository, held if it needs to be held, or distributed fresh. And as you've heard from some of the fantastic talks, that's sort of where several of them have gotten their tissue to do that. Also, so currently from the neuro-oncology clinics, we get biofluids. Right now, this is exclusively blood, but we're expanding this, and we'll be expanding this in collaboration with many of you to CSF as well. We have storage facilities so that anything that is banked can be stored as cold as possible. So in liquid nitrogen, we also have 2-80s for short-term storage. So the tissue bank was begun really with the insight of the neurosurgery department back in the 70s, and it has been acquiring and banking tissue in a very methodological way that allows people to go back and get those samples, and that sort of improved over time. And so because of that, we actually have quite a large archival collection, some just flash frozen tissue, but many with paired blood. But obviously the older patients and the older samples will not have the current therapies. And so within the last year and a half, this is the current archival. Now, remember, these are collected and banked. These does not reflect what we've actually distributed. And so that is additional material that is collected throughout the year. As you might expect, lung, melanoma, and breast are the three most common that we have archival in the bank. And these are stored as flash frozen tissues, which can be useful for a number of analyses, including single nucleus RNA sequencing, as Harish mentioned. So now I'd like to take a sort of a look at the second part of what the core does, and that's the cellular and molecular characterization of tissue. And so one of the goals is identification of cell types, very simplistic, but important. So for example, microglia and macrophages, as we heard about CD163 and IBO1, reactive astrogliosis or GFAP, in this case, this is adjacent to a metastatic carcinoma. Also characterization of the T cell response. So T cells looking at CD3, CD8, FOXP3, CD4, et cetera, in terms of helping to define what cell populations are there. And to sort of illustrate this to you, and also illustrate perhaps the importance of where you sample. And that's where some of the single cell technologies, as well as the spatial technologies, I think really are useful. Because here you can see two metastatic foci. This is intervening brain parenchyma. And you can see with CD3 T cells, you have an infiltrate that is adjacent to and within that surrounding brain parenchyma. And quite strikingly, in this particular breast carcinoma, you also have a really profound microglial macrophage response, as denoted by IBA1. But you can imagine, depending on where you sample in this lesion, you're going to get very different populations of cells. And I think that's an important thing to think about anytime you're doing sort of an analysis from bulk flash frozen tissue or fresh tissue as well. So in addition, we have markers that help with spatial localization. So for example, the identification of tertiary lymphoid structures in the brain really requires the identification of T cells as denoted by CD3, B cells by CD20, and microglia macrophages by IBA1 or other myeloid cell markers. In addition, one can look at co-localization of different components within the brain. So here's endothelial cells in green, you have IBA1 microglia macrophages in red, and then tumor cells in blue, again, sort of giving you another level of information on these tumors. Another important aspect that we're involved in is cell type specific expression. So for example, IBA1 microglia and macrophages, what other markers do they express? So in this particular case, we're looking at MHC class 1 as denoted by HLA-ABC. And in this case, we're looking at expression of class 2. And looking across different tumor subsets, as I'll give you an example in a couple of slides, these actually can be very different in different tumor types, as alluded to, I think, by Harisha's just talk, looking at different subsets of immune cells and sort of their function. This is in a BRAF mutant glioma, not a melanoma, looking at very high expression of class 1. And then this is an IDH-mutant tumor. So this is a tumor-specific antibody that we use, looking at activation of the PI3 kinase AKT mTOR pathway as denoted by phosphorylated 4-ABP1. Let's see. Sorry, my advancing slides has just stopped. Let's see. How strange. Okay, there we go. Another aspect we look at is, so those were all protein analyses. We also are able to look by in situ hybridization at RNA signals. And RNAscope is just a technology, but it's a very useful technology by ACD, which allows us to look at individual transcripts. So each dot here is actually a transcript. And so this allows us to identify, for example, here, tumor cells that express TERT. Here, this is in collaboration, and I should say this is in collaboration with Joe Costello's group. In this image here, you have a collaboration with Aaron Diaz's group, who has done a lot of single nucleus RNA sequencing as well. He had identified a potential ligand receptor pair, and this is demonstrating the receptor PTPRZ1 expressed on tumor cells and PTN, the ligand expressed on adjacent non-neoplastic cells, sort of lining this blood vessel here. So again, these kinds of techniques can help you sort of interpret, if you will, or confirm some of the analyses from other types of technologies. We also have been doing a lot with a quantitative whole slide imaging. I think the quantitative aspect to these analyses is essential. And so that's a big part of what we do as well now. And we've really developed that in the last couple of years, thanks to some fantastic members of the core. So what I'd like to do is take just a couple of minutes to give you some examples, some concrete examples where we've used these in 2 projects. And this was a project led by a very talented postdoc, Ezra Mohammed. So we know that in IDH-mutant gliomas, activation of the PI3 kinase AKT mTOR pathway has been implicated in progression. But when we look across a cohort, this is UCSF 500 data, looking at 60 patients with different grades of IDH-mutant glioma, both oligo and astrocytoma. We see an incidence, a frequency of about 22% of mutations that would be predicted to activate the pathway. But there had been some literature suggesting that the activation of the pathway at the protein level, so with post-translational modifications and phosphorylation, was higher. And so we wanted to investigate that. And so we developed a strategy where we identified tumor cells and then looked at activity of the pathway as denoted by phosphorylation of 3 different proteins in the pathway. And we took these same tumors, we took the same block actually that had been profiled by UCSF 500 and looked at protein phosphorylation. And what we found was quite striking in that almost 60%, 57% of these cases had quite robust evidence of activity of the pathway as denoted by phosphorylation of these epitopes, really suggesting that the mTOR signaling may be particularly important in these IDH-mutant glioma. And there's some interesting biology behind that, but sort of giving evidence of how even analysis of paraffin-embedded tissue can still be helpful in the right context with the right controls. This is a second example where we compared 2 different types of glioma. As you may know, in quite prevalent in the literature is the fact that the most common types of glioma have a very poor immune response. They have a very poor T cell response, but seem to have a very prominent microglial macrophage response. But we know, as a neuropathologist, we know that there's other tumor types which don't have this same pattern. And so we wanted to investigate that a bit further. And so Anupam Kumar is another very talented postdoctoral scholar who looked at this, comparing BRAF mutant glioma to IDH mutant glioma. And what using spatial proteomics demonstrated that there was quite striking difference in terms of an increased inflammatory response in these tumors, particularly a T cell response. And we expanded this in a cohort and did multiplex immunophenotyping and demonstrated that indeed the CD8 positive T cells were much higher. And looking at the transcriptional profiles, this is increased in BRAF mutant glioma, this is increased in IDH mutant glioma. We were able to demonstrate that there was increased antigen presentation and processing, in particular, increased expression of MHC class 1, and also increased expression of chemokine CXCL14, which we were quite interested in, and one of its putative receptors, CXCR4. And so by immunophenotyping, we could then confirm that indeed the tumor cells themselves had very high levels of expression of MHC class 1. We were able to also do this in patient-derived xenografts. We could demonstrate that these tumor cells, as denoted by the BRAF V600E mutation, had very high levels of expression of MHC class 1. And if we looked at expression of the ligand by RNA scope here, you can see that in a dense area of tumor, there was very high levels of CXCL14, confirming that the tumor cells themselves were expressing the ligand. And this is important because there's sort of a growing body of evidence, and this has been suggested in some of the prior talks, that the tumor itself plays a very important role in terms of shaping the immune response. And so understanding how the tumor does that, I think, is very important, particularly when one thinks of therapy, but also the biology of these tumors. And so this really spurred us to express CXCL14 in a murine immunocompetent model for GBM. And by doing this, we could dramatically increase the CD8 positive T cells present in these tumors. We could also prolong overall survival. And this prolonged survival was actually dependent on CD8 T cells. So I provide these as sort of examples where going back and forth between a number of different technologies can really help sort of expand what we can learn from these human tumor tissues. And so I'll just end with how can I work with the core? So many of you already know, and so this is preaching to the choir in some ways, but for those of you who don't, we sort of have three different routes, if you will. One is acquisition of de-identified biospecimens. So you email me or you submit a request online to the core. There's a tissue use committee which reviews all requests. And the Biorepository acts as an honest broker. And so we distribute de-identified samples to you, either archival or fresh from the operating room. And this is working very collaboratively with our neurosurgeons, who are an essential component of this. The second aspect is acquisition of biospecimens for research, where you have patient identification. So this is an IRB approved study. You still submit it so that we can review it. The tissue use committee can review it, approve it, and then we work closely with you because those patients have to be consented by your team. They get consented additionally for us so that we can handle the biospecimens, and then we can help you acquire those specimens in an approved way. And then the third interaction that folks may have with us is with our biomarker and pathology services. And so I just highlight the website here. Contact us button will send you right to the submit a request, or you can email myself or Annie Shea, who's the associate director of the core and really a critical component of that. And so with that, I'd like to thank all the members of the core. There's just a fantastic group of individuals who are really dedicated to this and helping investigators really get the most out of the samples that they're analyzing. The neurosurgeons here are just an absolutely fantastic group to work with and are an essential component of this process. Neuropathology as well, and then of course the patients and their families. And so with that and our funding, I'd be remiss to not thank the SPORE for its generous support to really support these kind of activities, because biobanking is not necessarily one of the things that can be well funded all the time. And so I'd like to thank them for that. And so with that, I'd like to thank you. And any questions, please feel free to email me with any questions if we don't have time today to go through those.